different patterns between normal and cancer samples. So the analysis provided by MS is very specific and sensitive. So that's why it is considered as a gold standard in disease diagnosis. However, the cost is very high compared to ELISA and other small kit method. Okay, last one, we want to see how the 2D SDSP image can develop our understanding about what happens during cancer disease. Okay. So 2D SDSP image can produce high number of protein spots, and usually each protein spot represent one individual proteins. It is rare to see two or three individual proteins in one protein spots. So that's why the resolution of 2D SDSP is very high, because almost all individual proteins can be separated. From the image, can compare the expression level only. We cannot compare, for example, the structure of amylase here. We can compare only expression level, and the significant difference between, for example, amylase expression level can tell us about a certain disease. For example, oral cancer disease. So if we identify all the proteins in normal and oral cancer samples, we can determine the number of proteins exclusively expressed in oral cancer sample, exclusively expressed in control samples, and also proteins shared by both conditions. So differentiating, categorizing the identified protein is important, because this can help us in identifying relevant strategy to treat oral cancer disease. Some treatments focus on proteins exclusively expressed in oral cancer disease, while the others focus on the proteins exclusively expressed in control samples. So after 2D SDSP and MS analysis, usually we come up with this kind of analysis. It is called a Venn diagram analysis. We can see the shared proteins and also exclusive proteins. Okay, from MS, we can see the unique mass spectropics. So the unique ones represent the abnormal expression of cancer biomarker. Okay. So the same approach can be used in other type of cancer. For example, breast cancer, and we can monitor the biomarker expression using MS analysis. Okay, any question regarding biomarker? Yeah, regarding examples of biomarker, you can read on your own. Too many biomarkers actually, so we just limit the biomarkers to those related to testicular tumors, hepatocellular carcinoma, and others. So if you can list down several of them I think would be sufficient to explain about the biomarkers. Okay, any question regarding biomarkers?
No, doctor.
Okay, my last word is that uh, to study the biomarkers, to use biomarkers in disease diagnosis or understanding the um, antimicrobial mechanism, for example, we need to have different set of samples. At least we have two set of samples. The first sample is control or normal. The second one is abnormal or patient sample. By comparing different samples, we can see the changes. And these changes could be seen either using SDS speech experiment, Western blotting, or MS analysis. If we do not use control or healthy sample or normal sample, we cannot determine whether biomarker is uh, changing or functional in diagnosing disease. So bear in mind that to use biomarker in diagnosis, we need to use at least two different samples, control and abnormal sample. Okay, that's all for today. Wassalamualaikum.
Waalaikumsalam. Thank you, Doctor.
Thank you,
Thank you,
doctor.
doctor.
Thank you,